Hello, everyone, and welcome to today's live broadcast, Examining Cancer with minION, Methylation and Structural Variation, presented by Winston Timp, PhD, Assistant Professor, Department of Biomedical Engineering, Johns Hopkins University. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Oxford Nanopore Technologies. For more information about our sponsor, visit nanoporetech.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the slide window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Timp. I will now turn the presentation over to him. Dr. Timp, the floor is yours.
Hi, thanks Christina for that kind introduction. So today I'm going to be telling you a little bit about our work in using the minION nanopore sequencer to try and characterize two specific elements of cancer. DNA methylation, specifically cytosine methylation, and structural variants, or areas of the genome that get disorganized in the process of carcinogenesis. At the top of the presentation, I'd like to disclose that I have two patents that have been licensed to Oxford Nanopore Technologies, whose technology I'll be talking about here and who are sponsoring this LabRoots presentation. So, to begin, I'd like to describe what is nanopore sequencing and when I say a nanopore sequence, what do I mean? So specifically in the context of this presentation, I'm describing the technology developed by Oxford Nanopore, which consists of a biological pore, specifically CSGG, as pictured in the bottom left here, which is a relatively small biological pore, less than 1 nanometer in diameter. When you thread DNA through the pore, this affects the current, as pictured in the middle there, and you can actually, using differences in the current, you can determine what the DNA sequence is. This is a really, the sequencer that they produce is quite small. It can fit in the palm of your hand. It plugs into a standard computer via USB port, and it can generate per nanopore flow cell on the order of 3 to 6 gigabases, or 3 to 6 billion bases of sequence data. How does the sequencer work? Well, the basic concept is similar to that of a Coulter counter. So the Coulter counter is this idea for counting cells and characterizing cell size. You can detect and you can characterize objects based on the blockage of a hole, in this case a nanopore, but it could be any size hole with the Coulter counter concept, and the current, the electrolytic current that's flowing through, the ionic current flowing through. Most nanopore work is either performed with ion channel proteins like alpha-hemolysin, MSPA, or CSGG, or it's in fabricated silicon nitride or graphene membranes. Most of the work that's done with nanopore sequencing nowadays is still focused on distinguishing exactly what's in the pore based on both the duration, how long things are staying in the pore, and the level of the current blockade events, what the current levels are. So to sequence DNA on the Oxford Nanopore instrument, we need to make what's called a sequencing library. To do that, we start with high molecular weight, nice long pieces of DNA. We break it up a little bit just to make things a little easier, and then we clean up the ends and then add specific sequencing adapters. These adapters contain a motor protein, which allow the DNA, the rate of DNA motion through the nanopore to be controlled. It also helps, there's also a tether that gets attached, which helps to bring the DNA down to the pore. When you add DNA to the nanopore flow cell, you want to get it to the pore as quickly as possible. And so if we tether it to, in this case, a bead that gets delivered to the pore effectively, then we're that much closer to the pore and we can get our results that much quicker. Pictured in the top right corner is an image from a typical, from the computer of an instrument connected to the Oxford Nanopore instrument. And what you see here is the sort of a typical response where you see pores, of the 512 pores available at any given time for sequencing, we see that 252 are in strands, so to speak. There's a strand of DNA actively going through the pore. That's the bright green. The slightly darker green color indicates pores that are open but don't have a piece of DNA in them right now. And then there are areas where there's zero current, so the pore is blocked or not operational, or the pores are saturated in either light blue or dark blue. So those are areas which aren't working so well. This is sort of looks like a typical one. So changing gears a bit, let me give you a brief description of what do I mean by structural variation. I said my talk at the top of the presentation was going to partially be about structural variation. Structural variation is an abnormality in a relatively large region of a chromosome, anything in the vicinity between 50 bases up to 3 megabases or 3 million bases. This is pervasive in cancer. It turns out that 50% of pancreatic ductal adenocarcinomas, or pancreas cancer, the most common type of pancreas cancers, have structural variations. They're common in tumor suppressor genes, in genes which normally keep cancer from progressing, like CDKN2A, P16, and SMAD4. So what this means is that when you get variations, you get chunks of these genes getting deleted, or aberrant chunks of chromosomes getting inserted into these genes, or duplications, or differences in variation, you're going to have problems because those genes aren't going to operate in a normal fashion, and that's partially how cancer can develop. Short read sequencing. So the dominant sequencing of the time of the common era is shorter reads, either through Thermo Fisher's ion torrent instrument or Illumina sequencing. Both of these read lengths are limited to on the order of hundreds of base pairs at the most, 2 by 300 or 400 base pair reads. This is great for detecting small mutations, but for resolving structural variations, it's kind of hard because structural variations tend to be in relatively repetitive areas of the genome, and you'd like to have a sequencing read that can stretch across the entire boundary of where you had the insertion to figure out the context of where that insertion is. Nanopore sequencing and to that extent PacBio sequencing are ideal for this. If you have a nice long read, you can stretch across an insertion, you can stretch across a deletion or a tandem duplication or an inversion, and you can get exactly the context and resolve that structural variant very effectively. So with the advent of long read sequencing technologies and the recent increase in yield of the long read sequencing technologies, we're at the forefront of being able to detect and characterize these structural variations that are occurring in cancer. One problem is that yield is going up, but it's still a little limited. Nanopore sequencing, for example, still has only about 3 to 5 gigabases per flow cell. For reference and context, the human genome, the human haploid genome, so one copy of each chromosome, would add up to something on the order of 3 gigabases. So that means you'd only have about 1 read in each region of the human genome. And if you had a heterogeneous sample that had some normal cells and some cancerous cells, you might have some trouble detecting it if you only had 1 read covering each region. You want to have higher depth so you had a probability of catching that structural variant. Luckily, there are methodologies to zoom in and capture only specific regions of the genome. I'm going to tell you a little bit about our efforts in one of these methods, specifically in adjuvant solution-based hybridization capture. So Adjuvant Technologies is a company which produces, among other things, hybridization capture baits, what's called baits, and kits. So let me walk you through this procedure. You start with genomic DNA. You then put on adapters. You sort of break it up and put on adapters to various sizes. Then you use, in the case of Agilent's kits, you use an RNA bait, which is a short oligonucleotide, which is antisense, which will hybridize, which will, the A's will match to T's, the C's will match to G's, and bind onto some of your genomic DNA strands. But it will only bind on in the areas that we care about. So we design our probes, our baits, very carefully so that they're only in regions around, say, for example, P16, a gene that we know has lots of structural variations in cancer. You then would use a magnetic bead, which will bind to these baits, and you can pull out with the magnetic bead just the DNA molecules that you actually care about. Then you can wash away the rest and just sequence what remains. And so now I can sequence in a targeted fashion just the parts of the genome I care about at relatively high depth. The trick was, when we tried to approach this, that most of these technologies are optimized for shorter reads and for fragmenting DNA to on the order of hundreds of bases. And we weren't sure how well it was going to work for longer molecules. So with Agilent, we wanted to develop this method to target something on the order of 2,000 bases long, 2 kb. We were being sort of conservative at first. Our first trial, we used tiled probes, so probes that just marched across our entire region in relatively dense tiles that would be close together. We targeted P16, which is and the region around it, basically 1.5 million bases of sequence in and around the P16 region. We used relatively low stringency probes, so probes that matched pretty well, but didn't match perfectly, or could match a bunch of different locations of the genome, because we wanted to make sure we wanted to pull down as much DNA as possible in this first round. But as a result, we found that there was a really low percentage on target. Only about 2.25% of our reads were actually in the region we wanted them to be. The rest of them were elsewhere in the human genome. So this was no good. We went back to the drawing board. And for trial 2, we didn't use tiling. Instead, we knew that we had big molecules we were trying to pull out, so we could keep our spaces pretty big because we only needed one probe per about 2,000 bases long, because the piece we pulled down would be 2,000 bases, so it would still stretch across. In this case, we actually were able to target, because we needed fewer probes, we could target 2 regions. So we targeted P16, CDK, and 2A, and we also targeted SMAD4. We used really high stringency probes, so probes that were really unique and sequences that you couldn't really find elsewhere in the human genome, to make sure that we would only get the regions, or hopefully only get the regions we cared about. We did this in consideration of known structural variation breakpoints. We then applied this to pancreatic ductal adenocarcinoma, again, the most common form of pancreas cancer, samples from Jim Eshelman's lab, a researcher here at Johns Hopkins. The result was that we got about 30% of our sequencing reads were on target, were right where we wanted to be. This was, as I mentioned, this was in collaboration with some of our associates at Agilent, specifically Josh Wang. So our targeted sequencing performance was actually quite good. Using here, I'm showing you here a control, which was a NA12878, which is a lymphoblast cell line, which has been highly characterized. It's one of these DNA samples that everybody likes to run their techniques on the first time, because we know almost everything about it. It's been sequenced by every technology you can imagine. We tried it with both Illumina sequencing and with Nanopore sequencing, and what's pictured in the top right is an image showing you the coverage, the number of reads we had per base that mapped to these regions. And you see that the sort of light blue rectangle indicates the region we wanted to capture for this SMAD4 region. What you see in the red is the Nanopore coverage, and what you see in blue is the Illumina coverage. And you see that they both echo each other pretty well, and the coverage is pretty uniform across the thing. It's jagged, yes, and there's regions where the coverage seems to drop out, but that's because we didn't have probes that were high stringency we could design in that region. The table below sort of shows you a summary of these results. For the Illumina data, we sequenced on the order of 4.4 million reads. These reads are short reads. 3.7 of those 4.4 million were on target. So our on-target percentage for Illumina sequencing was quite high, 85%. This was a fold enrichment of 640 fold enrichment over what you would expect normally from just throwing in the human genome, and gave us a coverage of about 113x. Nanopore sequencing, in contrast, wasn't as great in on-target percentage, only about 30% on target. And as you note, we only had 107,000 reads. This was from a slightly older Nanopore technology than is now available. You can get higher yields nowadays, but I wanted to present these results to show you guys. 30% on target, about 350x fold enrichment, which results in about 30x coverage. That's pretty good. That means we can get 30x coverage of this region. We can say with some confidence where structural variants and what's going on in this region. So we were pretty excited by that. And similarly, we did the same thing for the pancreatic ductal adenocarcinoma sample, and we got about the same on target percentage with a really good fold enrichment. And there, coverage was a little lower because we had fewer reads, but it was still quite good. So what can we do with this Nanopore data? Well, what we can do is we can look for structural variations. We can look for areas which are different. So using the NA12878 cell line, we then used our Nanopore data, aligned it, or matched up where it lay in the human genome. And then we tried to detect structural variations with a piece of software called Sniffles, which was developed by Mike Schatz's lab, who is of Johns Hopkins and Cold Spring Harbor. And Sniffles detected a tandem duplication here, as shown in the slightly darker red in the middle of the plot here. So I'll circle it. This is a similar duplication that was detected with deep PacBio sequencing data from the Genome in a Bottle project from Mount Sinai School of Medicine. This is a project where I mentioned earlier, NA12878 has been sequenced by every technology known to man. The PacBio data was also able, with its long reads, to detect this sequencing approach. So this duplication has been validated in NA12878, and we could pick it up with our targeted capture. Another example, but this is in a novel sample, the pancreatic ductal adenocarcinoma cell line. We found a new structural variation that we didn't see when we did Illumina sequencing of this sample using our Nanopore data. So what's pictured here, the y-axis shows individual reads as different rectangles, but you see in these reads here on the bottom that you have a big gap in the middle. That gap's actually a part of the genome that got deleted in this cancer sample. It's not everywhere. You see that these reads up here don't seem to have that same deletion. Remember, we have 2 copies of every gene, so this seems to be a heterozygous or a partial deletion. So either some of the cells in the sample had the deletion and some didn't, or perhaps one of the alleles had a deletion in this area and the other allele did not. So Sniffles and with the Sniffles software, with the design of probes that we had, doesn't detect all possible structural variations. What's pictured here is in red, the NA12878 coverage across this region of P16, a specific region of P16. And in green is the pancreatic ductal adenocarcinoma sample. And what you see here is that we detected a novel structural variant just by sort of observation, that you have 0 coverage in this region for the PDAC sample, whereas in the NA12878 we do have coverage. What this suggests is that that region was deleted, but the region was so large. Note that it is in excess of 400 kilobases long, so 400,000 bases long. We don't have any reads which happen to come up to the border. Note that right before you get to that border, we had a drop-off in coverage, coincidentally because of how our probes were designed. So we just didn't have any sequencing reads that happened to stretch across this deletion. So we did not cover the breakpoint, and so we could not detect this deletion with our data. We can pick it up by just looking at coverage, luckily, and there are methodologies to do that, but Sniffles is designed to only detect things that you find from breakpoints, at least the version we used was. So manual examination and software which looks for copy number variation or gaps in coverage can pick these up, and with our capture methodology, we can still see these. So taking a slight step to the side, typical short read sequencing is very accurate. It's 99.9% accurate in terms of on a single base level. Nanopore sequencing, as you may have heard, is not necessarily that accurate. The raw accuracy of base call data probably rests around 90%, 92%, depending on how you look at it, the quality of the DNA you're putting in, etc. But embedded in the electrical signal that you have from the Nanopore sequencing is more data. There's more information there. So what we want to do is we want to go back into the electrical signal and try and parse out and tease out what's there. And we think we'll be able to get a higher accuracy. We'll be able to call even down to the SNP level what the results are. This is software that was actually developed by Jared Simpson at the Ontario Institute for Cancer Research, one of our collaborators. He developed the software before we even started working together, specifically to be able to correct, to go back in and get better genome assemblies and be able to call even down to the SNP level what the data is, relying on partially on the actual electrical signal evolving from the nanopore sequencer. So what essentially happens is that you input the sequencing data and you use a hidden Markov model, which I'm not going to go into in great detail here, to determine what's the right answer. You know, as the DNA is evolving, as the electrical signal is coming off the pore, is it actually a G or is it really a C? Looking at the reference sequence of the human genome, we can actually determine what the genotype is, what the answer is, even down to the SNP level. So we tried. This has been applied to a public data set as published by Jane et al. Currently as a preprint in bioRxiv of NA12878 and had pretty high genotype accuracy at all sites, 99.2%. That's pretty good. And at variable sites where it's heterotypic, it was 94.8% accurate. So again, pretty good. We tried to apply it to our capture data as pictured here. So let me walk you through this slide. In the top left, you see a region of chromosome 9, which was part of our captured region. The purple squares at the very top there are the true somatic nuclear variations or sort of SNPs, areas of single base alteration. I also have pictured here the results we got from doing the Illumina sequencing capture of this region. You see that we got 6 of 6 right and no false. So the 6 indicates the number of true positives and the 0 indicates the number of false positives. So it didn't get anything wrong and it got all 6 right. Pre-nanopolish, just using the raw nanopore sequencing data, we only got 4 right, and we got 3 that aren't really there because of the nanopore sequencing's sort of error rate. But after going back to the raw electrical signal, we were able to correct some of these and look, not telling the nanopolish where the SNPs were. It was able to go in, correct the data, and look to see what the actual accurate sequence is, using the fact that we know what the reference is and we know where we should be looking. And we got actually 6 correct there and 2 incorrect. So that's pretty good. And looking at it over the entire region, as shown in the bottom right table, after nanopolish, we have a precision of about 93% in calling SNPs and sensitivity of about 26%. This is not that dissimilar from the Illumina data covering the same region. The sensitivity, and sensitivity refers to actually picking up. Precision says how many times you're right. Sensitivity says how many times you catch it at all. So we didn't really catch it some of the time, but that's again due to how we designed the probe. So we're going back and trying to do better design of probes to make sure we really capture the region in depth. But the important aspect to realize about this is that means that along these 1 to 2 kb long reads, we actually are going to get phased information. So shown in the top right is an image generated from IGV, which is a sequencing visualization software. And there, what you can see is sort of phased SNPs. So on the left, you see each row is a different sequencing read. You can see that we're calling this homozygous SNP, a difference from the reference human genome, accurately, that most of the reads after nanopolishing are called correctly as sort of orange, which is, I forget what letter that is, but it's a different letter in the reference. In contrast, you can look at this other area to the right where we have a heterozygous SNP. And you can see that we have areas where there is no SNP, and then on these first 2 areas, and then there is a SNP here on the right side. Similarly, we have areas where there's this SNP and this SNP, but nothing here. So we're actually getting phased information across stretches of the genome. This is amazing because this means for the first time we're going to be able to get long phased read information in specific targeted regions. Typically, this has to be imputed from correlation. It's usually not so hard because you only have 2 alleles, so you can figure it out from Illumina data. But it's nice that we can actually stretch across the entire region and get phased information because we have these long reads. And we're getting that, let me emphasize, we're getting that at the same time as we're getting the structural variant information. So we'll be able to identify what allele, if any, these structural variants are on we think with confidence. So closing the discussion about structural variation, I would now want to move to talking a little bit about epigenetics and methylation. So as somebody who works in epigenetics, I'm contractually obligated to show the figure here on the right, which is an epigraph from Waddington from 1940, giving us the historical definition of epigenetics, which is how genotype, how your genes, your genetic sequence interacts with the environment to create phenotype. So the analogy is that you have a ball rolling down a hill. So when you're a stem cell, you're sort of up here. As you roll down the hill, you become differentiated. The genotype sets up the hills and sets up the structure of the landscape. And the environment decides that you're over in this valley or in this valley, one or the other, and causes you to become lineage committed. You become a brain cell or a liver cell or a lung cell or a stomach cell. What valley the ball rolls into is not necessarily predetermined, but more of a stochastic behavior. The image I like to use to illustrate that is this cat, Rainbow, which is actually the first clone cat. And you can see that this cat doesn't look like the parent cat at all in terms of coloration. I know it's stochastic variegation, but the point here is that the variability you have in phenotype, there is some element of probability in determining what that is. A more modern definition of epigenetics involves, is usually set as heritable changes other than the straight genetic sequence, which is to say positive feedback loops, higher order structure. Where DNA is in the nucleus actually matters. If DNA is at the periphery of the nucleus versus in the center, DNA at the periphery usually isn't expressed, and DNA more in the center of the nucleus is more expressed. Chromatin organization, whether it's closed chromatin or whether it's more open chromatin so it can be accessed and expressed. And finally, DNA methylation, which is what we're going to talk about. In mammals, in humans, DNA methylation usually occurs in the context of CG dinucleotides. So it can be palindromic and then regulated after semi-conservative replication. The analogy I like to make is to a computer system where the DNA sequence is sort of like the hardware, the user input is like the environment, systems biology is like the running programs, and the epigenetics is like the RAM. It's like the active memory. It tells the cell, you're a brain cell, you're a brain cell, you're a stomach cell, you're a stomach cell. It's just constantly telling it to itself so it knows, even if it's taken out of environment, what it's supposed to be doing. So can we look at DNA methylation with nanopore sequencing? Well, two groups showed that you in fact could back in 2013. Jens Gundlach's group and Mark Atkinson's group demonstrated that with research pores, not with doctorate nanopore, that you were able to see changes in current evolving over time as from a methylated base, from methylated cytosine versus an unmethylated cytosine. So shown on the left, excuse me, shown on the left is the current for a methylated sequence in red versus the unmethylated sequence in black. You can see a relatively significant change in the current. And the same is shown over here on the right, where you can see the profile for a methylated cytosine versus an unmethylated cytosine. The current profile looks substantially different. So what we did in our recent paper that was published in the spring of this year in Nature Methods is with Jared Simpson, we implemented a classifier for detecting 5-methylcytosine using Oxford Nanopore data. So the first thing we did to do this was we measured distributions of current for different K-mers from E. coli. So nanopore sequencing works by detecting not one base at a time, but several bases at a time. So we looked at the currents that you see from running, for example, AGG-TCG through a pore versus TG and TCG-AGT through a pore. So the interesting base here is this C, the second to last C in that K-mer, and the second C in this K-mer. So what I want, and we did this with two different chemistries that Oxford had developed because the paper took a while to get out. So we started with the so-called version 7.3 pore, which has since been deprecated, and then we also performed this on the R9 pore, which a derivation of which is now in use, and as 9.4, and the 9.4 pore shows similar results to what I'm showing you here. So what I want you to note is that the unmethylated data current distributions are shown in red, and the methylated current distributions are shown in green. And what you can see is that for some K-mers, we see a pretty significant difference in current. So, for example, here we see that there's a big increase in the amount of current for methylated versus unmethylated K-mers, where this is statistically significant. You'd probably be able to pick this up pretty easily. In contrast, for this K-mer, the current difference is really small. When we looked at the R9 pore, which is the smaller pore that's sub 1 nanometer in terms of its aperture, we find that we still see a pretty big difference in this k-mer, although the distributions generally are wider. But the difference, and now we're even able to pick up a difference in this k-mer, although it's still pretty weak and not that significant. There's clearly a difference there. So this gave us lots of enthusiasm to move forward. We then, so then we applied what's called a hidden markup model to try and distinguish what was happening here. Essentially, what we do is we go through, because the nanopore is reading multiple bases at a time. We start with, for example, TAGTC, say. The nanopore then advances, allows the DNA to advance one base through. You then have to end up with either AGTCA, AGTCC, AGTCG, etc. And so you sort of have a guess at what this has to be, and then you also know what the current has to be. So you can combine those two probabilities to maximize sort of the joint probability, and you can trace the most probable path through this sort of network, through this hidden Markov chain, to figure out what the right answer must be. When we do that, considering whether it's more likely for a given position to be methylated or unmethylated, and we estimate what's called the log likelihood ratio. So we take the probability that it was methylated, and we divide that by the probability that it was unmethylated, and we take the log. If that ratio is more than 2.5, so if it's more than 200-fold likely that it was methylated than unmethylated, we call it methylated. If it's more than 200-fold likely that it's unmethylated, we call it as unmethylated. If it's between those, we say we don't know. This was sort of the threshold that we set for ourselves to give us a confidence that we really knew that it was methylated or not. So when we apply that to, again, our good friend NA12878 lymphoblast gDNA, we found that it correlated really well with the sort of gold standard for methylation characterization, which is whole genome bisulfite sequencing. So pictured here is the data for the 7.3 pore on the left and the R9 pore on the right for comparing NA12878 sequencing in CPG islands, in regions of dense CG, and what the overall level of methylation was in these regions for these samples. And you see we got a correlation of about 0.83 for 7.3 and 0.84 for R9. So realizing, of course, that most of these gene promoters are unmethylated in human samples. So our correlation is pretty good. It agrees with conventional data. Here's another piece of information showing that correlation. So published at the same time as us was a paper from Benedict Patton, where they showed they had a different methodology, and it was published back-to-back in the same issue. They have a different methodology for classifying methylation. So picture on the left is their methodology, on the right is ours, and you can see that both of them show very good correlation between bisulfite sequencing methylation levels and nanopore sequencing methylation levels. I also want to note, this is from, again, Mitten Jane's bioRxiv preprint, where they had dense, relatively dense, say in excess of 20x coverage of the NA128 dataset. This was part of a consortia where they just threw a lot of sequencing at that sample to get relatively deep sequencing. And you can see that we correlate really well. When we get the deeper sequencing levels, our correlation is quite good with the conventional bisulfite sequencing data. When we looked at our own NA12878 data, and we decided to try and bin it to compare it to transcriptional start sites, so we could sort of see if we see the same kinds of profile. Typically, as shown with the black line here, bisulfite data indicates that methylation dips as it gets close to a transcriptional start site, and then as it goes back into the gene body, it rises up again. As it goes further away from the transcriptional start site, upstream in the gene, it typically goes up again. We see that same kind of behavior for nanopore sequencing methylation data for both the 7.3 and the R9 pore. We also wanted to run some control samples. So we took some human DNA and we made all the CGs methylated enzymatically, and we made all the CGs unmethylated with PCR. And that's pictured here for the completely methylated and here for the completely unmethylated. When I say completely methylated, I really mean about 95% methylated as measured by bisulfite sequencing. We then measured how accurate are we actually calling this, how much of the time do we get the so-called completely methylated DNA right, and how much of the time do we get the completely unmethylated DNA right. So the 7.3 pore was 91% accurate. We were only confident about calling 68% of the CG sites because of the log likelihood threshold we were setting, but we got 91% accurate of those. For the R9 pore, we were 94% accurate at 77% of the sites. So this is pretty good. And pictured on the bottom right here is an ROC curve of that same data, sort of showing you our true positive versus false positive rate. And this is a pretty nice curve showing you that we're pretty confident in the accuracy of our methylation calls using nanopore sequencing. So I then want to sort of tell you briefly about our efforts in the regime of cancer sequencing with this methodology. So we wanted to try and apply this to cancer, but the yield, as I mentioned before, was still a little low. So we used an enzyme to cut up the DNA and kept the fragments that were between 3.5 and 6 kb. This allowed us to keep only about 12.5 megabases of the human genome as opposed to full 3 gigabases. So we could focus our sequencing in, giving us on the order of 10x coverage from the sequencing runs we did from these samples. We sequenced that fraction with both nanopore sequencing and with bisulfite Illumina sequencing. And that's pictured here on the left-hand side. So you can see that we see the same DMR differentially methylated region with bisulfite sequencing as we can also resolve with nanopore sequencing. And other than that, the profile looks pretty similar with bisulfite and nanopore. The absolute levels are a little bit different, but generally the profile looks similar. Pictured here on the right is an image, this is where it gets a little bit more exciting, is an image of individual nanopore sequencing reads that stretch across the same region as pictured on the left. So the left is a summary of, for all the reads, what's the percent methylated. But here what I've done is I've broken that up on the right, and I'm showing you, individual reads as each row, and the colored dots indicate if it's methylated and blue or unmethylated in red. And what I want to especially draw your attention to is this area here where the differentially methylated region is. So you can see in the normal samples, as you sort of expect in the more normal samples, which are the green MCF10A, which is a relatively normal-ish cancer cell line, and then the aggressive cancer cell line, the NDA NB231, which is in orange. So here, this top one is the normal cell line. You can see that a lot of this region seems to be unmethylated. It seems to have a lot of red dots. And we see that same thing over here in the percentage, and we see relatively unmethylated even in the bisulfite. When we go down to the cancer sample, you definitely see more blue, but what I want to draw your attention to is these reads here colored red. Note that it's not just that you're getting an increase in methylation here in the differentially methylated region. Instead, it looks like this entire read is pretty blue. The entire read got methylated. So that entire molecule, in a non-local way, got an increase in methylation. This might say something interesting about what's happening in the epigenome of cancer samples, in that there's entire stretches of the genome that are getting aberrantly methylated. And heretofore, it's been hard to see that because we only had small reads, so we could only capture, this piece and then this piece, and we didn't know that these patterns were correlated over long stretches. So I'm sort of excited about this. This is obviously preliminary, and we have to keep working on it, but I think we're going to be able to get some information out about that. Moreover, with Nanopolish, we can get the SNPs and the methylation on the same long read. So we can see, this is again drawn from the high coverage NA12878 public data set. We can see areas which have a SNP, a heterotypic SNP, so shown here. And you can see that this top haplotype, which has the SNP, seems to have a lot of methylation. So methylation is either these black rectangles or these sort of, as pictured here, or a sort of lighter gray rectangle as pictured here. And you can see that for this, when you have this SNP, this allele of the NA12878 seems like it was highly methylated. Whereas this allele down here at the bottom does not seem to have that same high level methylation. You can see that whereas like this region here is methylated, this region isn't. This region is all very methylated. This region isn't. So we're seeing allele-specific methylation. Your dad's copy of the chromosome might have been methylated, whereas your mom's wasn't, or vice versa. So we can finally really get at, using these long reads, we might be able to get at long-range patterns of allele-specific methylation. So we're excited about that. Well, and then another direction we're going with this methylation work is we're going to expand this to not just CGs. The way we trained and the way we taught our model right now was to look pretty much only at CG methylation. We want to expand that. We also want to expand to looking at non-methylcytosine methylation. So we want to look at GATC, because we think that we're going to be able to use that for exogenous labeling of DNA and be able to label like where DNA is in the nucleus and whether it's open or closed chromatin. So we're excited about that. And we wanted to also apply this to real clinical samples, to get not just cell lines. We want to take out some tissue and sequence that with Nanopore. So I'd like to acknowledge some of the people who have worked on this project. People from my own lab, myself, Rachel Workman, Stephanie Howe, Yunfan Fan, and Isaac Lee. People from Jim Eshelman's lab who have worked with us on detecting and talking to us about the structural variations and providing some of the samples, Jim himself, and Alexis Norris, who has since graduated. And then, of course, our great collaborators at the Ontario Institute for Cancer Research, Jared Simpson, leading the efforts there, who has done the development of the software, of the Nanopolish software, so we can actually classify this methylation data. And then some of people working with him, Zuzuate, Matai David, and Dersi. And then finally, I'd like to also thank our collaborators at Agilent Technologies who have helped us with the capture methodology and some of that work. I'd also like to acknowledge some of our funding sources, both from within Johns Hopkins internally and also from the NHGRI for sequencing technology development. Thanks, and I'm happy to take questions.
Thank you, Dr. Timp, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question of the day asks, can you detect methylation with targeted capture method?
That's a great question. So the question was, can you detect methylation with the targeted capture method? So short answer, no, currently. And the reason is because when you're capturing such a small part of the genome, you know, a megabase or so, that's 1/1000th of the genome. When we put, you know, a microgram or so of DNA into that, that means we're only saving 1,000th at best if we had 100% efficiency. So we'd only have something like a nanogram of DNA back out. That's just not enough with our current methods to sequence. So we have to do some PCR, some polymerase chain reactions and amplification. What that means is that you've eliminated the methylation marks, and so we can't do a native methylation. We need to put in sort of raw DNA right from the cells to be able to look at the methylation marks. If you do PCR, the methylation gets erased. So because of limitations in our capture methodology right now, it's not possible. If we could put in more material, or if we could get our library preparations more efficient so we could sequence from, say, a nanogram, we could do it. So I'm confident it will be possible in the future, but right now it's not yet possible.
Thank you for your answer. Our second question of the day asks, can you capture larger molecules than 2 kb?
Great question. So the answer is yes. We have already shown in work I didn't present today that we can capture out to 5 kb, 5,000 bases long. And we're still striving to get it longer. We think there's going to be some limit where it's going to become too difficult to successfully capture and keep our on-target percentage, keep the number of things that map to the right area, capture the right DNA. It's going to become hard as we get longer and longer. But and there's also going to be diminishing returns from how long we really need to capture. But we think we can definitely get longer than 2. We're confident we can get 5. We're hoping to get 10 kb long. Thanks for the question.
Thank you for the answer. Since we're running a little short on time today, we'll only have time for one last question. And this question asks, can you detect 5-hydroxymethylcytosine with your methylation classifier?
That's a great question. So hydroxymethylcytosine, for those who may not know, is another type of mark that has recently been very interesting because it may be part of the loss of methylation pathway. It's also frequently found in neurons and in stem cells. So it's a mark that's very interesting and currently hard to classify with conventional methods. I expect that we will be able to find that, and in fact, in the paper that was published at the same time as ours, the back-to-back paper from UCSC, they showed that in fact you can detect hydroxymethylcytosine with Oxford Nanopore data. So the answer is yes. Our classifier doesn't yet do it. We're working on getting a nice training methodology to do that. But the answer is definitely yes. It's possible. We just need to train. The trick is generating the right training set so we can be able to say with confidence where the hydroxymethylcytosine is, and if a molecule does have hydroxymethylcytosine, methylcytosine, or normal cytosine. We think that pretty much, more globally, we think that pretty much any modification to DNA is potentially detectable because it's going to change the molecule that's actually in the pore and therefore change the current. So we think we're going to be able to see all sorts of different modifications to DNA or even RNA. It's just a matter of training and figuring out what kind of currents you see from those different modifications. Thanks for the question.
Well, thank you for all your answers. I would like to once again thank Dr. Timp for his presentation. I would also like to thank LabRoots and Oxford Nanopore Technologies for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us here at LabRoots, and we hope to see you again soon. Goodbye.